Hello everybody and welcome to today's webinar. My name's Emily Perry and I'm in charge of the Ensembl outreach team, which means I run a small team that's involved in teaching and training in various different ways on using Ensembl. We're also the people who will answer your emails if you ever email Ensembl. So I'm going to be talking today about genomic features that regulate gene expression with Ensembl. I'm here from Ensembl, which is a genome browser, and genome browsers are fantastic ways to look at genomes. Genomes are in themselves pretty useless as types of information. They not particularly informative when all you're looking at is a big long string of A's, T's, G's and C's. So genome browsers are a method of rationalizing that data, which we do by plotting different kinds of data against the genome. So what we have in Ensembl, we have genes plot against the genomes of over 100 species. We have gene trees. We compare all those genes to each other. We have what we call the regulatory build, and I'm going to talk about that. That's the main focus of today's webinar, but that incorporates data from places like ENCODE. We have ways of looking at variation data. We allow you to display your own data. I'm going to talk briefly about Biomart, which is a data export tool, and also programmatic access via our APIs. And one of the things that's really cool about Ensembl is that we are completely open source. So all our tools and software and everything that goes into our website is freely available. You can use it, you can download it, you can run it yourself. It can get quite complicated, but you can do it and access all our data. Ensembl is accessible at various different scales, so you can look at a gene at a time or a regulatory feature or a variant. You can look at lists of them or you can look at the whole genome at once. So we have the browser, which is what we're mostly going to be focusing on today, but I will mention Biomart and the REST API, which can both be used to look at groups of data as well as the variant effect predictor. I'll also talk a bit about the Perl API, which can be used to look at, can scale up or scale down to whatever scale you're interested in. And also I'll point you to our FTP site where we have some files that you can download and get the kinds of data types that I'll be talking about on a genome-wide scale. We do release data on in a 2 to 3 month cycle. So we're currently on Ensembl 96. Which came out in March, and we're coming towards the end of this cycle. But what we've been doing over the past couple of months is we've been adding in things like new genome assemblies, updates to the underlying software. Excuse me. We update the gene sets, we compare all our new genes, we update variation data, we update our regulation data, we change and update our interfaces, and later this month, hopefully, fingers crossed, we will have Ensembl 97 out before the end of the month. So what I want to talk to you about with gene expression in Ensembl is epigenetics in gene regulation. Hopefully this is a bit of revision, but I just want to talk about how it impacts on what we can display in Ensembl. I want to talk about methods in epigenetics, and then I want to talk about what we use, where we get the data from, and then what we do with it in order to predict these features that regulate gene expression. So epigenetics is the study of heritable genetic changes without changes in the gene sequence, which is known to regulate gene expression. And of course epigenomics is that, but on a genome-wide scale. We know that epigenetic change is linked to cell differentiation, and that cells which carry out different functions, which are morphologically different, which express different genes, have different epigenomes. So they have different things, different histone modifications across their genome. They have different methylation across their genome, and they have different things like transcription factors active and binding. So the methods of gene regulation that we use in Ensembl, that we use to predict our regions that regulate gene expression, we use histone modification data, which we get from ChIP-seq. We use transcription factor binding data, also from ChIP-seq, and I'm including in that RNA polymerase and CTCF. RNA polymerase being the ultimate transcription factor. And we use open chromatin data, which we get from DNA sensitivity. I know that FAIR is sort of taking over from that a bit, but we don't have any of that data, I don't think, yet. So we're talking about things like promoters and enhancers and things. So if we imagine a gene, and upstream of the gene we have a promoter, and in order for RNA polymerase to come along and transcribe that gene, it can't do it by itself. It needs transcription factors bound to make that happen. But sometimes it also needs the help of a region further away, a dysregulatory element or an enhancer. And then we get more transcription factors bound, and we get this crimping of the genome to bring these regions together, and then our RNA polymerase can bind. So we need transcription factor binding for transcription, and the combinations of epigenetic marks affect how transcription factors can bind. Now, transcription factors bind to transcription factor motifs. These are short sequences that attract transcription factors, and we use motifs that come from the ChIP-seq project. Now, the motif I'm showing you here on this slide is a CTCF motif, and it's pretty big actually as far as motifs go. Most of them are only about 8 bases long, and this demonstrates this is a short sequence that the transcription factor binds to, and at each position with the sequence within the sequence you can have, there are some bases, so position 4 here is always a G. If you've got anything other than a G at that position, your transcription factor is not going to bind. But if we look at position 7 and 8 here, these are both ambiguous. You can have options for different motifs, and some motifs will be stronger than others. So if we look at position 2 here, where we've got G, that's a stronger transcription factor because we observe that more frequently. A would be a weaker transcription, would cause a weaker motif than the G. So we have transcription factor motifs from the ChIP-seq project, and we have these plotted all over the genome. And we have them plotted on the genome, and we will indicate if they are known to be bound to that transcription factor from ChIP-seq data as well. We also have histone modifications. As I said, hopefully this is a bit of revision. So histone modifications, histones are these massive octamer proteins which have the DNA wrapped around them twice. They have these big dangly tails which are rich in arginines and lysines, and these arginines and lysines can be methylated or acetylated, and the methylation or acetylation of these of the tails affects transcription, and they're described in this format, and you'll see this format in Ensembl. So we've got H3K36Me3, so this would be histone 3, this green one here. K36 is lysine 36, so it's the 36th amino acid, not the 36th lysine. And Me3 means it's trimethylated, but it can also be monomethylated or dimethylated. And this is just the histone code. So some histone modifications we know promote transcription. Some histone modifications we know dampen down and prevent transcription. Others just don't occur. We don't observe them. And these are some of the major histone modifications that we use in our analyses of where our active and inactive promoters and things are. So ChIP-seq, I'm just going to quickly go over ChIP-seq as a method because it impacts on the data that we can see compared with what's actually happening biologically. So biologically, what we have is a DNA binding protein, which is bound to DNA. And if it's a transcription factor, it frequently has a very small footprint. It can be sort of 20 bases or something like that. We do cross-linking, so we introduce a covalent bond. And when I say we, I don't mean we at Ensembl. We don't do any wet science. People like ENCODE and Blueprint and whatnot do this. So they'll introduce a covalent bond that binds the DNA binding protein to the DNA. They shear the genome, and they use a random method for this, such as sonication. And as you can see in this very crude sketch, This particular region where we've shared the genome, it's the DNA binding proteins roughly in the middle of the fragment, but over here we've got one where the DNA binding protein is actually at the end of the fragment, and this is what happens when you use a random method. You use an antibody to your protein of interest to pull it out, so now you only have the fragments that are bound to your protein of interest. You remove the crosslinks, you wash, you sequence the fragments, you map them back to the genome, you see where your transcription vector is bound. The reason I mention this is because when you look at transcription factor motifs versus ChIP-seq peaks, they are very, very different in size. So if we imagine this CTCF binding motif, which is actually a very large motif, and our ChIP-seq sonication fragment, you can see that it's about 10 times the size of it. So you've already got a massive expansion of what your fragment is. Your read length is about 50 bases. You're going to be using something like Illumina sequencing for this. And so what you end up with is our motif in the middle, quite small, but up to 400 bases either side of that motif, you will have 50 base reads sort of accumulating over it in this peak. So you'll generally call the peak as being some point in the middle of it, but it won't necessarily perfectly represent where this motif is. And that's one of the things you've got to kind of keep in mind when you're looking at this kind of data. We also look at open and closed chromatin. And just to point out, we do the ChIP-seq data. We have that for transcription factor motifs and histone modifications. Transcription factors, not motifs. Motifs are just sequence features. So we also have open and closed chromatin data because we know that open chromatin, where the histones are spaced out, is more transcriptionally active. Where it's closed and all bunched up, that's less transcriptionally active. So we use DNA's hypersensitivity, but we could also use things like FAIR data, where you use DNAs to digest the DNA, and you get a lot of small fragments. You discard all the bits that are still attached to histones, and these loose bits you purify, you sequence, you compare to the reference, and you determine where you had this open chromatin. So these are the data that we're working with in Ensembl regulation. So what Ensembl regulation is trying to do is go from this state of saying we've got tons and tons, and we really are talking thousands of experiments of ChIP-seq data on our histone modifications and our transcription factors, and actually going to something more simplified and saying this is where the promoter is, this is where the enhancer is, and is it active or inactive in different cell types. So we use data from a number of different sources. In human, we have data from ENCODE, Roadmap Epigenomics, and Blueprint Epigenome. We have ChIP-seq and histone modifications and transcription factors and DNA sensitivity from both ENCODE and Roadmap. Blueprint, we only have the histone modifications and the DNAs. For mouse, we have ENCODE data for a number of cultured cell lines as well. So it's cultured cell lines from ENCODE and Roadmap. Blueprint, we're looking at primary cells from the hematopoietic cell lineage. We don't display all the cell types that these groups have studied. When we do our build, we need to have CTCF binding, which is not available for Blueprint, and we need to have histone 3 lysine for trimethylation, histone 3 K27 trimethylation, histone 3 K36 trimethylation, and we process these data. So we take the data from the raw data and we predict where we've got promoters, enhancers, insulators, and which cell types they're active in. So if we imagine this is again very simplified version of what the data would look like from a single cell type. What we have, so our squiggly like a wiggle track here is showing us the number of reads for a particular experiment, and these blocks here are giving us the peaks. They're giving us the point where we've said, yeah, this is significant. These are reads that we think are relevant. And we find that we get clumps where things are happening. These are the regions that we think are actually doing something. So this would be a region that we think is having some kind of activity. And we say, well, we're seeing this cluster of these two different transcription factors, these three histone modifications together. Is it doing anything? So then we look across the whole genome. So we use actually machine learning to do this. We look across the whole genome and we identify all the regions where we see similar clusters to what we saw before. Now, because of how the techniques work, you don't get these perfectly neat clusters where everything's in a line. It's all a bit messy. You don't always get everything. But you can see you get similar patterns across the genome. And if we know that a couple of the locations where we see this pattern are promoters, we can then extrapolate and say, well, everywhere where we see this pattern is probably also a promoter. But it's not so simple as saying, well, that's what a promoter looks like, because we'll find another pattern which turns out to be a promoter, and we'll find another pattern which also turns out to be a promoter. So you have loads and loads of different patterns across the genome, and we train this machine learning to recognize, to identify these patterns and to recognize them. And it produces what's called segmentation. So across, in a single cell type, it can then say, this part of the genome is doing this part of the genome is doing that, based purely on this pattern finding. Now, one of the cool things about this pattern finding is it's actually completely blind. So the algorithm doesn't know what the patterns it's looking at are. So we don't tell it things like this is an activating histone modification. It just knows this thing, wherever I see this thing, it matches this pattern. And we give the pattern the name. And so you can then go back and say, well, these activating, that we've got activating modifications at activating segments and vice versa. So this kind of proves that our machine learning is working. And it's something that we're improving on continually. As we add more cell types into the algorithm, more data into the algorithm, our pattern recognition gets better and better. So we can then line up all the data from all the cell types across the genome next to each other, and we can say, well, we've got more patterns. We've identified that this region, so this red region over here, has promoter activity. And what we actually say is, well, this whole red region is a promoter, and this whole yellow region is something else, and this whole blue region is something else. And it's active or inactive in the different cell types. So we say that this is where the promoter is, and then we'll tell you whether it's active or inactive. So we use this kind of candy stripe notation to mean inactive. We actually have different levels of activity that we will describe something as. So if we call something active, so if we say, we use the promoter as the example because it's the easiest one to follow. If we've labeled something in the color that goes with it, so that in the case of the promoter that happens to be red, we say that it's active. It's got transcription factor binding and histone modifications which are consistent with promoting gene expression. We also have this status which is called poised, which means it's ready to be activated. For a promoter, that usually means it's got everything except RNA polymerase. It's got everything it needs. It just needs a trigger, and it will go. We also have inactive, so it just lacks the stuff that would make it active, and repressed, which means you have transcription factors and modifications which are preventing activity. So this would be things like, if you compare the histone modifications, active would have activating histone modifications, inactive would have no histone modifications, and repressed would have repressive histone modifications. There's also the option of no data, which means that particular cell type, there isn't the stuff we need to be able to call it. So if there's a cell type where there's no CTCF data, CTCF binding sites would be called as no data. So we've covered quite a significant amount of the genome. So we have transcription start sites, promoters, which also have flanks to them, enhancers, CTCF binding sites, and also what we call unannotated transcription factor binding sites, which are regions where we have transcription factor binding, but we don't really know what else it's doing. And actually that covers about 10% of the genome, has got this kind of annotation on it. The question everybody asks when I show them this data is, are these promoters real? Well, they're based on real biological data. We're not just making them up. We're not just using patterns in the sequence. It's not like when people say, oh, well, the 1,000 bases upstream of the gene is a promoter. This is actually taking into account the epigenome. We have strong evidence to suggest they're doing what we think they are, but most of them haven't been experimentally validated. Nobody has gone through and cloned every single one of our promoters alongside a gene and seen if it promotes transcription. That would be a massive experiment. So we don't have that. And as we gain more data, we feel like our pipeline is being refined and improved and giving more and more accurate data. Another thing we don't do is we don't actually link promoters and enhancers and insulators to genes. We show you where they are and we allow you to make your own inferences. For promoters, this is generally fairly obvious. The big promoter at the 5' end of your gene is almost certainly the promoter for your gene. Wouldn't absolutely depend on that because you can get things like the bidirectional promoters, but it probably is. We also don't link it to gene expression at the moment. So we have cell line specific regulation data and tissue specific expression data. Recommend you turn on data like that and look at it together, but we haven't made that link. It is still incredibly complex and in its relative infancy, and there's no sort of comprehensive database that links all the promoters to their genes, that links all the enhancers to their genes. Another thing we have, which is really cool, is variation data linked to gene expression. So we have expression QTLs from GTEx. So you can see if a variant up or down regulates genes in different tissues. So if you can see a variant that upregulates a gene, it falls in one of Ensemble's regulatory features, you can make a reasonable inference that that regulatory feature, even if it's a trans-acting one such as an enhancer, you can make a reasonable inference that that it's regulating that gene. So enough of just talking through slides. We're now going to have a look at a region in Ensembl. We're going to find regulatory features. We're going to see what cell types they're active in and what evidence there is to show this. So I'm going to go to the Ensembl genome browser. Is that showing? Yes, excellent. And I'm going to search for a gene. So the gene I'm going to search for is LIMD2. And I'm going to click on the link here to the region of the gene rather than the gene name. Because when we're looking at regulatory features, we're usually looking at surrounding regions. So this is our gene. In case you're not already familiar with the way things are displayed in Ensembl, this is actually a negative strand gene, which I can tell because I've got little arrows pointing in the direction of transcription. I've got valley-shaped introns, and it's also shown below this blue bar. So we've got a gene just here which is above the blue bar. That's a positively stranded gene. So that means if I'm looking for a promoter of this gene, I'm going to be looking over at this side of the screen. And if I scroll down the page, I actually see this promoter is massive. It actually covers the gene. And it's quite an interesting thing that we've discovered is that we actually can see epigenomic activity related to the activity of the gene in the promoters frequently spanning the gene itself and sometimes the neighboring genes. So I know it's a promoter because I know the color codes because I work with it all the time. That's interesting. The legend is not showing. I'm going to have to report that to our people. There should be a legend that says that red means promoter and that this cyan colour here means CTCF binding site. If I click on it, I will see that it's a promoter, and I'll show you with the CTCF binding site. It tells me it's a CTCF binding site, but I will report that to our team. But I can see that it's a promoter, and it's got a stable identifier, which I can click on. I've also got coordinates for it. So I'm going to click on the identifier, and that takes me to a page which gives me information about it. Now it's just taking a moment to load. So it shows me the context of this promoter. So as you can see, it spans most of this gene and also part of this neighbouring gene. In this view, I can see the motifs, the transcription factor binding motifs in this track down here. All of the grey ones are motifs where we know that the sequence is there, the sort of little 8 or 10 base sequence is there, but it's not, we haven't got ChIP-seq binding data that confirms that the transcription factor actually binds there. But the black ones indicate where they are bound. And if I scroll down the page, I have a list of cell types. And it tells me which cell types this promoter happens to be active in. This is quite an active one, so I've got 121 out of 123 cell lines it's active, and 2 of them it's inactive, so I'm just going to remember these 2 cell types so I can use that for some comparison. But as you can see, I've got a lot of data, a big long list of cell types where it's active. If I want to find out more, I can go into this details by cell type. And I can use this to choose actually which data I can see. So if I click on this configure cell/tissue, I can choose which cells, which tissues I actually want to see. So I remember there was a cell type called H1 trophoblast, which was inactive. So I'm going to select that one as a cell type of interest. And I'm going to pick a couple of others. So I'm going to pick this HUVEC and HepG2 as well at random. So I can pick which cell type I want to find by clicking through this alphabet ribbon, but I can also search for a cell type of interest. So if I wanted to look at something that was in, say, here we go, neuronal progenitor, by typing NEU, I could do that way, and I could tick it. Having done that, it says I've now picked 3 cell types. If I click into experiments, I can say, right, I want to see all the histone modifications. So I can do a select all. I want to see all the transcription factor binding sites. I want to see all the open chromatin. I can search this in exactly the same way as I could with the cells and tissues. So I've got the search. I've got the ribbon for the transcription factors. I don't need the ribbon for the histones, because that would be pointless because they all start with H. And then I can click configure track display. And this allows me to choose, it shows me what data is actually available. So you can see that actually for a couple of these cell types, for the H1 trophoblast and the HUVEC cells, if I scroll across here, there's a bunch of transcription factors that are not available. And I think this is because the H1 trophoblast is actually a cell type that was studied by Blueprint, and they didn't do transcription factors. We've got loads of data for the HepG2s. And I can decide whether I want to see this or not. So the first sort of stage is filtering down how I want to see it. I can still make further edits to what I want to see. So I can choose that I don't want to see this particular modification. So I can get rid of everything with that modification. It might be more clear if I scroll. Here we go. I can say I want to get rid of everything with this modification, or I can say I want to get rid of everything for this cell type rather than cell of the, or I can get this cell as cell of the table. But I'm going to leave it with everything, and I'm going to hit view tracks. And now this is going to plot all of this data. So all of this ChIP-seq data and DNA sensitivity is going to show me what there is for these different cell types, and it's going to show me their activity as well. So I can compare why the particular call was made, that this particular promoter was active or inactive in these different cell types. It's being a bit slow. I should maybe have loaded this before I started the webinar, but never mind. Here we go. So you can see in this H1 trophoblast, so I've got this candy stripe pattern, which tells me that the promoter is inactive. It's also worth noting that it shows me the blue, as we said, CTCF binding site, which I can remind myself of here, and that's actually no data for the H1 trophoblast. But you can see I've got peaks of binding of a number of different kinds of features. So I've got no DNase 1 sensitivity. It's closed chromatin, big indicator that it's inactive. I've got a bunch of histone modifications. But if I look at and I can see both the sort of peaks, the wiggly lines, and the blocks, which I could have changed. If I go back into my configure, I can change this so that I can see the wiggly lines, the signal, and the blocks. Both. I've got it set to both, but I could choose to change it to see one or the other if I wanted to. And now it thinks it's loading again. That's helpful. Even though nothing has changed. If I look at my HepG2, I've obviously got a bunch more data, but I've got some more features that indicate activity. And similarly, the HUVEC, I've got a bit less data again, but I've got more data that indicates activity. We've got an interesting feature here. It still thinks it's loading, which is why it's a bit pastely coloured. You can see here we've got a black line in my features in HepG2. It's gone, it's proper colours. And no matching black line here in the H1 trophoblast cells. If I click on that black line, I find out that this is a transcription factor binding motif. So we've got a CTCF motif and an ELF1, ELF2 motif within this region. And the fact that it's black indicates to me that it is actually bound. So if I scroll down and I've got CTCF here, you can see that CTCF is actually bound at this region. If I find my ELVs, where's my... Oh, so that's ELF1. I don't think it was just ELF1 and ELF2. I think it was more because it had like dots after it. Now it's going to take a while. We won't worry about that. Actually, we will, because I want to click on the thing and show you. So if I click on one of the motifs, it shows me, okay, so there's a whole bunch of transcription factors that share this motif. So I can see that probably one of these, it might take me a few minutes to find it, but one of these will actually be bound at this region. But I can click on it and I can see what the motif actually looks like. I can also do the same if I go back to my location view that I looked at earlier. I can manipulate that, and over a larger scale, if I just click on this blue button, configure this page. If you're not familiar with Ensembl, this is a menu that allows you to add all kinds of data onto this view. But if I go into features by cell tissue, you can see I've got the same interface that I had earlier, and it's actually ported the stuff across that I looked at in the other view. So I can look at over a larger scale if I wanted to see the binding over a number of genes. If I was looking at a cluster like the Hox cluster or something like that, I could look at the region over a large region over a number of different regulatory features as opposed to just one. So that's what I wanted to show you for looking at kind of single regulatory features. I want to mention some of the bulk access methods of getting to these data. So one of the tools we have is Biomark. If you've used Biomark before, you'll know that it's a really useful tool which allows you to to kind of specify and customize tables of data that you want to download. So you choose the database and species that you want to look at. You then filter your data, so you're choosing basically how many rows your table has. So does your table, if you've got a row for each promoter, you're choosing which promoters are actually going to have rows in the table. You get your attributes, which is your columns in the table, what data you actually want to know about all of those regulatory features you find, and you can get it as results. So if you go into Biomart, when you choose your data set, you can go into Ensembl regulation, and there are actually a number of different databases you can use. So there's binding motifs, there's other regulatory regions, which gets you things like phantom. Regulatory evidence, which will get you stuff like the actual ChIP-seq, and regulatory features, which will get you things like the promoters and microRNA target regions. So an example of how you would do this is you would use the data set of the Ensembl human regulatory build. You would filter by, say, your region of interest and your feature type. You would get its identifier, the cell types it's active in, as an example, and a table. So let's just do a quick query. If I just close this and go into Biomart in the top blue bar, and I choose Ensembl regulation and regulatory features. The query I'm about to do is massive and stupid, and would probably Biomart wouldn't be too chuffed with me for doing it, but I'm going to do it anyway because otherwise it involves me coming up with arbitrary numbers. Oh, come on, load for me. If it doesn't load in the next minute, I will just carry on and pretend this never happened. But I'm going to filter it, so I'm going to get all of the promoters on a small chromosome. I'll go with chromosome 21, and I'll get their locations and whether they're active, which cell types they're active in. No, I think I'll give up on this. If you are not familiar with Biomart, if you've not used it before, there's a tutorial video on using Biomart here that you can get to. And it's just a case of exploring the filters and seeing what you can find. I will go back. You can also get the regulatory data through our FTP site. So our FTP site basically have massive flat files of all our genome-wide data. So you can get the regulatory features and their activity as GFF files. If you download the slides, these should all be links in the PDF you get. If they're not, then let me know and I will find a way to get that information to you. So you can get the ChIP-seq peaks and signal. So you can get the cheeks as GFF, peaks as GFF, big bed and bed, and you can get the signal as big wigs. You can get the segmentation as big bed, and you can also get the transcription vector motifs as GFF, and that's by chromosome. There are a lot of them, as you'll have seen by just looking at that tiny region. So these are going to be massive files, but if you want to analyze the data on a genome-wide scale or do sort of similar kinds of analyses yourself, you can do that. We also have a REST API if you want to work programmatically. We have a number of different endpoints in our REST API. I'm not going to go into too much detail about what REST APIs are, but if you know about REST APIs, you can get data, so you can do things like put in a regulatory feature ID, and it will give you a bunch of data, including things like its activity in different cell types. You can also do things, we've got these, what we call the overlap endpoints, so you can put in a region and you can find all the regulatory features in those regions. We have an online course. If you go onto EBI Train Online, you'll find there one of the online courses is using the REST API course, and there's an online course which actually has you finding promoters in the region upstream of a cell type using some of these endpoints, so you can link these data together. So we do have tons and tons of training available online. You're possibly already aware of EBI Train Online, which is how you came across this webinar. I cannot push enough how good EBI Train Online is. We have some Ensembl courses on there. There are loads of other bioinformatics courses as well. They are great. We have a YouTube channel. You may have already seen the EBI YouTube channel, which has a number of training videos. We also have one in Ensembl, which we replicate on Youku as well. So if you're ever in China and can't get to YouTube, you can use Youku instead. And you can email us anything that you're stuck with Ensembl, anything I've mentioned in this webinar, anything that you come across Ensembl. Otherwise, please do send us emails to helpdesk@ensembl.org. You can also follow us on social media. And we can come to you and teach courses. So we can teach a course at your institute for free. In high-income countries, we will ask you to pay the trainer's expenses, but in low-income countries, we have money to pay for that. If you send us an email, again, helpdesk@ensembl.org, we can come and do what we call a browser course. So we do a course aimed at wet lab scientists and clinical geneticists and molecular biologists, people like that, which shows you how to do all the point-and-click interface and the data that's available. We also have a REST API course aimed more at bioinformaticians and kind of wet lab wannabe bioinformaticians. And we can also, mainly in the low to middle income countries, we can train people to deliver the training that we do as well. There's more information on this if you go to training.ensembl.org. We also have publications. If you use our data, please do cite us. So the Ensembl 2019 publication is the kind of general one. But if you want to know more about how we produce this regulatory data that I've talked about today, this paper, the Ensembl Regulatory Build by Daniel Zabino and others, is really, really good. If you want to learn more about how you actually access it, another paper also by Daniel Zabino, but I'm on this one, so you should cite that one because I get the publication, I get the citations. No, you should cite everything. That's also available as well, and that describes more about how you get to the data. This is Ensembl at our retreat last year. This is again us and the people in our team. Well, thank you very much for attending. If there is something you think of after the webinar, please just send us an email and we will get back to you. Thank you all for attending.